discussion of the lymphatic system and the immune system. I hope you're doing okay today. So this is something I would normally do in 3 class periods, so I'm going to break it up into 3 parts. So the first part, we're going to talk about exactly that, the lymphatic system. What is it? What does it do? What is in lymph anyway? The lymphoid organs, and then I'm going to cut it off. Then when I come back, I'm going to talk about the non-specific immune system. So this is the part of your immune system that protects you, but it doesn't get better over time, and it has no memory. And then I'll come back for the 3rd round, and then we'll talk about the specific or adaptive immune system. And this is the part that gets better over time. We'll talk about different types of immunity, allergies, and autoimmune diseases. So go ahead, kick back, relax, and let's do this. So this is a picture of the lymphatic system. It's shown in green. Lymph is not actually green, but they had to pick a color, so they did, and they pretty much keep this consistent with all anatomy textbooks. It is a system of vessels, cells, and organs that, one, carry the excess fluid back to the bloodstream, so we kind of already talked about that function, but while it's returning that fluid back to the venous circulation, it is also going to be filtering. And the idea is that this is where immune surveillance occurs. And so this lymph is going to be filtered, and we will mount an immune response to eliminate those pathogens, those disease-causing organisms from our body, hopefully before they make us sick. And so we've got some vessels, we've got some organs, we've got some cells, and I'll just tell you right now, the parts are semi-independent of one another. So it's not one of those cohesive systems where they're all connected. So I've given you this picture in your notes or table in your notes, and so this is kind of where we're headed. So we've got the lymphatic system, which has that sort of sewer system function, but then it's also the immune system. And so the immune system is a complex collection of barriers. So our first line of defense is going to be our skin and our mucous membranes. We'll talk about the different cells and the different organs that are involved. But once again, we're trying to destroy and neutralize pathogens that would otherwise cause disease or death. And so hopefully by the end of these 3 episodes, you will understand that picture. So what makes up the lymphatic system? I'll tell you. It is a network of vessels that are closely associated with the cardiovascular system. Difference is this is a one-way system and there is no pump to it, but it's going to begin in the peripheral tissues and end at the junctions with the subclavian veins. The fluid inside of those vessels is called lymph. It's very similar to plasma. And once again, that's that excess tissue fluid that it leaked out of the capillaries and wasn't returned on that venular side, so that interstitial overflow. We have some organs, and like I said, they're not connected to one another. The red bone marrow has a role in making the white blood cells. We'll talk about the role of the thymus, the lymph nodes, the spleen, and something called MALT, mucosa-associated lymphoid tissue. So we'll talk about payers, patches, appendix, and tonsils. So these pretty much are more the immune part, not the sewer part of the lymphatic system. The cells of the immune system, we know that they're white blood cells. We talked about some of them already, but notice lymph is in the word and lymphocyte is in this word. And so it's going to be these lymphocytes, the T cells and the B cells. They're going to circulate between the blood, the interstitial or tissue fluid, lymphatic vessels, and lymph nodes, all the while defending our body against infection. So the B lymphocytes, their job is what's called humoral immunity. They make protective proteins called antibodies. When they become activated to do so, that is when they become the effector part, and so they're called plasma cells. So an activated B cell doing its job is called a plasma cell. And so they're the ones secreting those large quantities of antibodies, and we will discuss in depth how that occurs. T lymphocytes, there's one population that secretes chemical messengers that's going to help the B cells become plasma cells. But the effector T cell is actually going to kill our virally infected cells and tumor cells by cell-to-cell contact. So they're responsible for our cellular immunity. Natural killer cells, they are a large granular lymphocyte. They are part of the innate immune system, so they don't have memory like T lymphocytes, but they kind of do the same thing. They're going to destroy virally infected cells and cancer cells. We discussed neutrophils, eosinophils, basophils. We talked about monocytes and macrophages. There's another type of cell that comes from the monocytes called a dendritic cell. It has pretty much the same function as a macrophage, except it is, if you want to think about this, a professional antigen presenting cell. It is going to be this cell that is going to talk to our lymphocytes and then activate our adaptive immune response. Another cell type, mast cells, and there they are right here. The mast cells are very similar to basophil cells in that they have granules of histamine. And so the mast cells and the basophil cells are going to stimulate what's called inflammation. These are involved in allergies or acute hypersensitivities, and we'll discuss that at the end of episode 3. And of course, we already talked about our neutrophils and their roles as the first responder, highly phagocytic, and they're seen in acute infection and in pus. So it's not just the lymphocytes, we have those non-specific white blood cells as well. So lymphatic system, one of its functions is that fluid balance. And so it's going to pick up that excess fluid away from the interstitial spaces and transport it back to the bloodstream. So remember, we saw this picture. We know that on that arterial side, hydrostatic pressure is the largest pressure delivering nutrients out into the interstitial. So we've got that push out, bulk flow out on that arterial side. We've got osmotic pressure as the greatest force on the venial side, pulling things in. But we know that we leave some fluid out here because the force pulling in is not as great as the force out. And so it is that lymphatic system's job to pick up the remaining 15% and return it to the bloodstream. That's roughly about 3 liters a day. You kind of need that to occur. We also know the second role is for immune defense. So screen for, remove foreign substances, and protect against pathogens and disease. This function is kind of an oddball function, and it's going to come up again in Unit 4. So this is showing the small intestine, which is where all our nutrients are absorbed. It is an organ with a high surface area, so it has villi and microvilli. But these little fingers are called villi, and this is a villus. In each villus, this thing right here, notice it's green, it is what's called a lacteal. So the third function of the lymphatic system is lipid or fat absorption from the gastrointestinal tract. Fats can't go into the bloodstream because plasma is mostly water, we learned that. So they get a protein carrier and they are absorbed into the lacteals, and then they'll go into those subclavian veins into circulation. So its role is in absorbing lipids, glycerol, fatty acids, cholesterol, and those fat-soluble vitamins A, D, E, and K. So we saw a version of this picture when we did the cardiovascular system. We talked about vessels. And so we are going to begin here in the lymphatic capillaries, and we are going to follow the pathway that lymph takes till it gets returned to that venous system. So it is a one-way system. And once that tissue fluid enters those vessels, that's when we call it lymph. So lymphatic capillaries are very similar to blood capillaries. They are just one cell thick, so they only have a tunica intima. The differences between them, though, they are incredibly permeable. So more permeable than sinusoidals. They have their cells that are kind of going to overlap each other, kind of like roofing shingles overlap one another. And they are going to have these fibers. And what's going to happen is when the fluid out here accumulates, that increases pressure, and these are going to swing open like little doors. So they're really, really permeable. Anything that's out here can kind of fit inside of there, and then the doors will close. They also have mini valves that allow for this one-way entry and to prevent backflow. Lymphatic capillaries are going to be interlaced among the arterioles and venules of the circulatory system. There are some places where they will not be found. You do not have any lymphatic capillaries in bone marrow. We know that there's the blood-brain barrier. You don't have any in the central nervous system, bone tissue, you won't have any teeth, cornea, that sort of thing. But everywhere else, there's going to be lymphatic capillaries associated with the cardiovascular system. So the capillaries, when they merge to become lymphatic vessels, they're going to be very similar to veins. They're going to have thinner walls. They're going to have more valves because this is a one-way system and there is no pump. So that's even lower pressure than we had in the veins. They're going to have the same three tunics as veins. So once again, the endothelial liner, so tunica intima. There's some smooth muscle associated with it, so the tunica media. And then the connective tissue outer binding layer, the tunica externa. Larger vessels lead to lymph nodes, and then to larger vessels, lymphatic trunks and lymphatic ducts. So we're going to follow that. So we have capillaries, collecting vessels. We got some lymph nodes we'll talk about in a minute. And then we have lymphatic trunks. Trunks are going to drain lymph from the lymphatic vessels, and they're particularly, they get named for where they are. So the jugular veins are going to be in the neck, subclavian underneath the clavicles. Broncho refers to lung, mediastinal, so in that mediastinum area, collecting drainage from the lungs. Lumbar, lower back, and intestinal. So they just sort of give them names of the trunks. And then from the lymphatic trunks, we're going to go to two lymphatic ducts. Okay, so the lymphatic system is not symmetrical, just like the cardiovascular system is not symmetrical. And I kind of, if you're a picture person, I gave you this little pie diagram. So one of the collecting ducts is called the right lymphatic duct, and it's kind of the slacker. It is only receiving lymphatic drainage from the right upper appendage, the right thoracic region, and the right side of the head and the neck. And so it's a short duct, and it enters right here in that right subclavian vein, right before it becomes the brachiocephalic vein. The other duct is by far the largest, and so does the thoracic duct. And depending upon how tall you are, but it's going to basically go from your abdominal region through the mediastinum up to that left subclavian vein, so roughly about 18 inches. It is by far the bigger duct and receiving the most of that lymphatic drainage. And what it's going to receive, so from the two bottom appendages, the abdominal region, it's going to get all the drainage from the gastrointestinal tract, the upper left appendage, left side of the head and the neck. So clearly you can see it's the biggest. This area right here is the expanded portion, so this is the proximal part, so the beginning of that thoracic duct. It is called the cisterna chyli. So a cistern is like a well or a reservoir, and chyle refers to this milky fluid which contains those dietary fats, those triglycerides, lipids, and proteins that were picked up and absorbed from the diet. And so it's going to pick that up from the gastrointestinal tract, and then it gets added into venous circulation. Pop over here for a second. I just kind of wanted to show you why they kind of named it milky. Think fat. You've seen fat on meat, maybe fat floating in water, but it kind of has that whitish appearance. So what can happen is if there is damage to this thoracic duct, you get basically this leakage of this fluid. And so it's called a chylothorax, so pleural effusion. And so this is just that drained lymph that was going to enter. So I just kind of wanted to show you what it looked like. So the cisterna chyli is that proximal part of the thoracic duct. It is by far the biggest duct, and then it's going to return that fluid to the venous system right there at the left subclavian vein. So lymph nodes, lymphatic capillaries, afferent arriving lymphatic vessels. We haven't talked about lymph nodes yet, and then it's going to lead the lymph node through efferent lymphatic vessels. Then it's going to lymphatic trunks, collecting ducts, and then into the subclavian veins, because the purpose is to pick up that fluid and return it to our blood supply. All right, so once that fluid gets picked up by those lymphatic capillaries, that's when it's called lymph. So hey, what is it? It's a lot like plasma. So it's going to be mostly water. It's going to have ions, some small proteins. We know the lacteals in the small intestine are going to pick up fat, so meal dependent. Also, since these little mini flaps open up so big, we're going to get cellular debris, It can pick up virus particles. It can pick up bacteria, so any sort of pathogens that are out there in the tissue fluid. Also cancer cells. So cancer is unregulated cell growth, and when these cells become cancerous, they often will lose their cellular attachments. And when they lose their cellular attachments, they start to wander, and they can get picked up by the lymphatic system, and then they can travel and cause secondary tumors. So when cancer moves or spreads, it's called metastasis. And so that's how cancer spreads. It's going to use the lymphatic system. So just for example, breast cancer can metastasize and move to the lung and cause tumors in lung tissue. Also, what's going to be in our lymphatic system are lymphocytes, and other white blood cells, so our leukocytes, our immune fighters. So just kind of reminding you, so we've got fluid out here in the interstitium. We're going to get more out than we get in, and so it's going to be those 3 liters per day. They're going to get picked up by the lymphatic system and returned, because what do we want? We want even. All right, so that was Pearl Jam. I had to come up with something for lymph flow. There you go, even flow. So what happens if there's excess fluid out there? I'm going to tell you that lymph moves really, really slowly because, once again, there isn't a pump. So when there's excess fluid out in the interstitium, that basically is the definition of swelling. So edema means swelling. So it's a temporary accumulation of excess tissue fluid. It can be due to trauma, so injury, or due to infection. And so it is going to be the lymphatic system's job to pick up that excess fluid and return it to the bloodstream. So I'm going to show you a lymph node, or an artist's rendition of a lymph node, in just a little bit. But I want to show you this picture. So this is a lymph node. So you have hundreds of lymph nodes scattered throughout your body, and they're often here's lymphatic vessels, and they're often going to be next to blood vessels. So once again, so you have blood vessels, and then you see your lymphatic vessels. So lymphatic system, cardiovascular system, very close association. There can be a special type of swelling. It's called lymphedema. And so this is going to be persistent swelling due to the obstruction of lymph movement. And so this can occur particularly after lymph nodes have been removed. We'll talk about breast cancer, how often lymph nodes are going to be removed, those sentinel nodes, to see how far or if that cancer has spread. There's also a parasite that causes elephantiasis, which can also block lymph flow. But think about it, if you block up the sewer, you're going to get excess fluid accumulation, so swelling. So how does lymph move if there's no heart? Guess what? Same way it does in the veins. So we have one-way valves to prevent backflow. The vessels, just like veins, they're going to be situated around skeletal muscles. So the more a person moves, the better their lymph return, and also the better their immune system. I have a research background, and I worked in a molecular immunology lab, and one of our many projects was the effect of exercise on the immune system. And actually, mild exercise, not hardcore workout when you are coming down with something. So it's sort of in that prodromal phase when you're feeling just kind of crappy. It's good to take a walk because you can help your immune system fight that infection. So it helps move lymph through the lymphatic system. Deep lymph vessels typically will travel by arteries, so that throbbing of the artery, so the pulse of the artery, will also help move things. Once again, we're also going to use our respiratory pump. And then too, that smooth muscle in that middle layer in those larger lymphatic vessels can also constrict. And so, yep, there is no pump to that one-way meandering system. So let's talk about the organs. So there's primary lymphoid organs. So they're called primary because this is where lymphocytes, our B cells and T cells, where lymphocyte stem cells divide and mature. So we learned when we discussed blood that all blood cells, formed elements, are made in the bone marrow, particularly red bone marrow. In the bone marrow, the B cells, the B lymphocytes, are going to stay there and undergo all of their development. T cells, T lymphocytes, so kind of pop down here with me, they're formed in the red bone marrow. They're going to be released as immature thymocytes, and they are going to go to the thymus. So when we discussed endocrine, I said I would bring this one up. So the thymus gland is where T cells are going to go to mature. Once they mature, and they're going to get educated there, then they will leave the thymus and then they will circulate. When the B cells mature, they will leave the bone marrow. And so both the T cells and the B lymphocytes, they're going to circulate between blood, tissue fluid, and lymph. The thymus gland, I'm just going to throw this out, it does not mount an immune response. It's just for education and maturation of those T cells. So here we go. This is just another nice picture. So we have primary lymphoid organs, so the red bone marrow and the thymus. The other lymphoid organs are called secondary, so lymph nodes and spleen, mucosa-associated lymphatic tissues and tonsils and things like that, because it's going to be in the secondary lymphoid organs. That's where our body is basically performing immune surveillance, and then if we have a pathogen, then activation. So it's showing you the origin of both T lymphocytes and B lymphocytes is in the bone marrow. T cells, so T and T cells go to the thymus. When they leave the thymus, they are mature, although they are called naive because they haven't met their antigen. They have not been stimulated yet. But then the T cells are going to go seed lymphoid organs. So it's showing you a lymph node here. B cells, B lymphocytes, they're made in the bone marrow. They mature in the bone marrow, and then they too are going to leave and they're going to circulate. B cells can do their job from those secondary lymphoid organs. So they don't circulate as much as the T cells. When they encounter their antigen, and so an antigen is anything that stimulates the immune system and mounts an immune response, so this is something that's foreign to you, they're going to encounter their antigen in those secondary lymphoid organs. And then once they encounter their antigen, that's when they become activated, and then we're going to get proliferation and differentiation into our effector cells, so plasma cells to make antibodies, and our cytotoxic T cells to kill our cells gone bad, virus-infected cells or tumor cells. So let's talk about the thymus. So the thymus, remember, it's right above the heart in the mediastinum. It is a bilobed organ that's going to secrete a collection of hormones that I'm going to collectively call thymocytes. They are going to drive the development of the thymocytes to develop into mature T lymphocytes. The thymus is the site of T cell maturation and education. Yes, it's like a school. This is not an organ where we will be fighting infection. I'm going to tell you right now, the majority of the cells that go to the thymus never leave because T cell education is brutal. You think your program's tough, right? Only about 2 to 5% are actually going to make it. The thing is, these cells have the ability to come alongside our body cells and kill them. So we kind of want to make sure they're not crazy assassins. So they have to be self-tolerant and immunocompetent. So they have to recognize self because they need to determine self from non-self. And they have to tolerate all of our self-antigens, so not mount immune response. And so that's where they're going to learn to do that. Other weird things about the thymus. So the thymus is most active during childhood. So this is showing you a thymus in a child, and then this is showing you a thymus in an adult. I will tell you right now, I don't have a model that actually has a thymus on it. So you can see this is showing you a thymus gland, a cross-section during childhood, and this is what it looks like in adulthood. And so this is a gland that's going to atrophy as a person ages. So it grows until someone's in adolescence, and then it begins to atrophy. And so it's called thymic involution, but basically this functioning tissue here that's involved in that T cell education and maturation is going to become connective tissue and fat. So it's going to be adipose connective tissue. So that's what, when something atrophies, that's usually what is replacing that functioning tissue, connective tissue and fat. So what's happening in that magical place, the thymus? Okay, so the first round is called positive selection. And so these T cells, they have to recognize self. And all of the cells have what's called an MHC, major histocompatibility complex. And so it is a glycoprotein on the surface of cells. And so they have to be able to recognize that barcode. So you got your MHC. It is going to be genetic. It's through recombination, so there's many different combinations. Only identical twins are going to have the same MHC. This is what has to be matched if someone's going to donate tissues to somebody. So that's, take that word apart, major, it's important. Histo-tissue compatibility complex. And so like I said, you think about it like a personal barcode. And so the first step, as those thymocytes, they enter the thymus directed by these thymus hormones, is can you recognize our self-antigen? And so they have to be able to recognize. So if they don't recognize it, then they go through apoptosis. So can you imagine if you fail the test and then you had to die? Once those cells, they recognize self, then the second thing is called negative selection. Then these cells are going to be presented with self-antigen, so self-proteins. And if they bind to that self-antigen and recognize it, then they die. Zip, you're out of here. Because that means they would mount an immune response against self. So they have to recognize the barcode, recognize you from something foreign, but not get too super excited by it to where they would mount an immune response. So fail to recognize self-antigens result in survival and continued maturation. So it's rough. This is a really messy picture, but it's just another way you can read through this on your own. So it's showing you that they come from the bone marrow. They're going to first enter that outer core, the cortex of the thymus. And so that's where they go through that positive selection. Do they recognize that major histocompatibility complex? So if they pass their first test, they get 2 cell markers, like it's kind of like 2 little graduation hats. And there's 2 flavors of these cell markers. So CD8 and CD4 stands for cell determinant or cluster differentiation 4 and cluster differentiate because, hey, all lymphocytes look all right, am I right? So they have these cell markers, and so you can tell one type of cell from the other type of cell. So then they go through negative selection, and that's when they're going to be presented with self-antigens. And the idea here is that they can't bind to them. We don't want them to bind to them and get excited. And so that's going to happen in the middle, the medulla of the thymus. If they pass that test, then they're either going to get 1 cell marker, either a CD4, and that means that they are going to become a mature T helper cell, which we'll talk about later when we talk about the specific immune system. Or they become a CD8 cell, and those CD8 cells, those are going to be the cell killers, so they're called cytotoxic T cells. And then they're going to be released to the lymphatic system. So those secondary lymphoid organs, these are organs where lymphocytes mount an immune response. So we'll talk about lymph nodes, we're going to specifically talk about the spleen, and then what's collectively called mucosa-associated lymphoid tissue. So things like Peyer's patches, appendix, TONSILS. So let's talk about lymph nodes. And so I showed you one, but this is an artist's rendition of a lymph node. Doesn't look a thing like it, does it? The 3 functions of a lymph node, they are going to filter foreign material from the lymph. So pathogens, cellular debris, so they have a disposal function. Since lymph nodes are going to be packed with immune surveillance cells, so packed with those B lymphocytes to make antibodies, and those T lymphocytes that are specific attack killers. And we also have our macrophages and our dendritic cells, so those phagocytic white blood cells. So we're going to have immune surveillance, and if a lymphocyte meets its antigen, so the one it's specific and destined to respond to, this is also a site of lymphocyte production. So we're going to get proliferation. So I didn't really lie to you when I said all blood cells come from the red bone marrow, because they do, but lymphocytes are special in that they can go through mitosis and they can divide. So lymph nodes are a site of lymphocyte production. So if we look at the structure of a lymph node, we are going to have a capsule. So we have, it's going to be connective tissue. And this dense connective tissue is going to wall off this lymph node. And we have these 1, 2, 3, 4, 5. We're going to have many afferent lymphatic vessels. So lymph's going to flow in, but notice we only have 2 efferent vessels at this little dimpled place called the hilum. And so think of merging traffic in Houston. Lymph is going to move very slowly through this lymph node. So we have it walled off by the trabeculae, and then the type of tissue that makes up a lymph node is that reticular connective tissue. I'll show you a picture of it in a minute in case you don't remember that from your histology, but think of it like it's a network. And this is going to be where the white blood cells are. Particularly, we've got these germinal centers, which are going to have those B cells. We're going to have T cells, so T lymphocytes. We're going to have macrophages and dendritic cells. But the goal as that lymph is being filtered is to basically sift it and see if there's any potential pathogens that we need to fight or mount an immune response to. So this is that reticular tissue. So those white blood cells and lymphocytes, macrophages, dendritic cells, they're going to hang out on these reticular connective tissue fibers. And so lymphoid organs are going to be made up of reticular connective tissue. Just right structure for the job. So yeah, so these are lymphoid follicles, and other parts of the body. So when we talk about mucosa-associated lymphatic tissue, they also have lymphoid follicles. But at the center of them, we have the germinal centers, which have those rapidly dividing B cells. We're also going to have T cells, dendritic cells, and macrophages. And then those B cells, they can become activated to plasma cells, and then they can just release those antibodies, those protective proteins. There are places in the body where you have aggregates of lymph nodes. Like I said, you have hundreds of lymph nodes scattered throughout your body, but there's places where there are little aggregates of them where they are superficial and they can be palpated, they can be felt. And so in the cervical region, so in the neck region, you probably have felt these. If you felt like you were getting an upper respiratory infection, sometimes your lymph nodes are going to swell. Underneath the arm, so the axillary area, and then also in the groin, so the inguinal area. When a woman has breast cancer, they will do a biopsy, they'll remove that cancerous tissue, and then they may also take some of these axillary lymph nodes. And that's how they determine what stage of breast cancer someone has. If they find those cancerous cells that have broken free in those sentinel lymph nodes, they'll remove them from the axillary region. Just kind of hold that thought. Okay, so let's kick back and watch the tale of, yes, Black Death. I know, I sounded a little too excited about that, didn't I?
Imagine if half the people in your neighborhood, your city, or even your whole country were wiped out. It might sound like something out of an apocalyptic horror film, but it actually happened in the 14th century during a disease outbreak known as the Black Death. Spreading from China through Asia, the Middle East, Africa, and Europe, the devastating epidemic destroyed as much as 1/5 of the world's population, killing nearly 50% of Europeans in just 4 years. One of the most fascinating and puzzling things about the Black Death is that the illness itself was not a new phenomenon, but one that has affected humans for centuries. DNA analysis of bone and tooth samples from this period, as well as an earlier epidemic known as the Plague of Justinian in 541 CE, has revealed that both were caused by Yersinia pestis, the same bacterium that causes bubonic plague today. What this means is that the same disease caused by the same pathogen can behave and spread very differently throughout history. Even before the use of antibiotics, the deadliest outbreaks in modern times, such as the ones that occurred in early 20th century India, killed no more than 3% of the population. Modern instances of plague also tend to remain localized or travel slowly, as they are spread by rodent fleas. But the medieval Black Death, which spread like wildfire, was most likely communicated directly from one person to another. And because genetic comparisons of ancient and modern strains of Yersinia pestis have not revealed any significantly functional genetic differences, the key to why the earlier outbreak was so much deadlier must lie not in the parasite, but the host. For about 300 years during the High Middle Ages, a warmer climate and agricultural improvements had led to explosive population growth throughout Europe. But with so many new mouths to feed, the end of this warm period spelled disaster. High fertility rates combined with reduced harvests meant the land could no longer support its population, while the abundant supply of labor kept wages low. As a result, most Europeans in the early 14th century experienced a steady decline in living standards, marked by famine, poverty, and poor health, leaving them vulnerable to infection. And indeed, the skeletal remains of Black Death victims found in London show telltale signs of malnutrition and prior illness. The destruction caused by the Black Death changed humanity in two important ways. On a societal level, the rapid loss of population led to important changes in Europe's economic conditions. With more food to go around, as well as more land and better pay for the surviving farmers and workers, people began to eat better and live longer, as studies of London cemeteries have shown. Higher living standards also brought an increase in social mobility, weakening feudalism, and eventually leading to political reforms. But the plague also had an important biological impact. The sudden death of so many of the most frail and vulnerable people left behind a population with a significantly different gene pool, including genes that may have helped survivors resist the disease. And because such mutations often confer immunities to multiple pathogens that work in similar ways, research to discover the genetic consequences of the Black Death has the potential to be hugely beneficial. Today, the threat of an epidemic on the scale of the Black Death has been largely eliminated thanks to antibiotics. But the bubonic plague continues to kill a few thousand people worldwide every year. And the recent emergence of a drug-resistant strain threatens the return of darker times. Learning more about the causes and effects of the Black Death is important, not just for understanding how our world has been shaped by the past. It may also help save us from a similar nightmare in the future.
Okay, so there's a couple things in that video. Hopefully you saw those little black bumps. Those were buboes. That's where bubonic plague got its name. So those were where they were superficial. They showed it in the neck, underneath the arms, and in the inguinal area, so in the groin area. Buboes are infected lymph nodes that are actually filled with pus. So the bacteria that cause the Black Death, Yersinia pestis, they could actually grow and live in those lymph nodes. I'm setting this sort of in your mind that your ability to respond or to fight a particular disease is going to be genetic. So the majority of the people alive today are relatives of persons that were exposed to and survived the plague. So yes, congratulations. I'm going to tell you that your ability to fight infection is because of your genes, not what disease you're being challenged with. So just kind of tuck that one away. So lymph nodes, because they are draining lymph from a particular area, if that is, so think about in the cervical region, so think about from your throat, so your upper respiratory tract. So the pathogens or virus particles, bacteria, anything that was in that area is going to get filtered in these lymph nodes. If it is something foreign that can cause disease, then those lymphocytes are going to respond. And so they are going to go through proliferation, and then they're going to do their job. So that sign, when lymph nodes are swollen, that means that those lymph nodes are draining sites of infection. So they're swollen with bacteria, debris, and those increased numbers of lymphocytes. All right. So the spleen is the largest lymphoid organ, so purely lymphoid organ. It filters blood. There's two parts to it, red pulp and white pulp. And I'll show you a picture of a fresh spleen so it'll make more sense. But it sort of helps remember the functions. So the spleen filters blood. That red pulp, so the part looks red, is because the function of the spleen is to remove worn out red blood cells. Remember, they only live for 120 days. Platelets only live for like 10 days. And then any sort of cellular debris. And then two, of course, we're recyclers, so we're going to recycle all of those breakdown products from hemoglobin. The spleen also stores blood, so it has open capillaries, so sinuses. And so it's storing for us excess red blood cells, platelets, and monocytes that the spleen can release these when we need them. So if we've been cut, and then two, when there is an infection. White pulp is aggregates of white blood cells, so cuffs surrounding central arteries. And so once again, we have white blood cells, we're going to have lymphocytes, that their job there is immune surveillance and to respond to blood-borne pathogens, because lymph nodes filter lymph, spleen filters blood. Big difference. The spleen, it does have a capsule, but it is not a very hardy capsule in the sense that if the spleen gets ruptured because of its large blood supply, so roughly about 1/5 of blood can go to the spleen at one time, usually what they're going to do is they will just remove the spleen, so it's called a splenectomy. If there is some of the spleen that can be saved, the spleen can regenerate, but the risk is that that person is going to bleed out, so hypovolemic shock, organ damage, and then death. So often they're just going to remove that spleen because it's very fragile. They can't really sew it up and repair it. It's too mushy of an organ. So the spleen, remember, it's in the upper left quadrant. It's tucked up underneath the diaphragm. It's going to be behind the stomach. And it's showing you this big, large splenic artery. And then you have the splenic vein that would be going away from it. See the pancreas. So like I said, it's going to be that upper right quadrant. Big thing too, it has follicles. So it has a similar function to lymph nodes, but like I said, instead it's filtering blood instead of lymph. So it's going to store blood for us, removal of worn out red blood cells, so the red blood cell graveyard, old platelets, it's going to store monocytes for us and platelets. And so just kind of remember those functions. So this is when they looked at a fresh spleen. They said, oh, look, red pulp, because, yeah, red blood cells and storing blood there. And then the white pulp would be aggregates of those white blood cells, particularly the lymphocytes. So that makes a little more sense. Other lymphoid tissue is called MALT, mucosa-associated lymphoid tissue. And so this is just going to be just below the mucosa. There's going to be lymphoid follicles. So we're going to have once again, our immune surveillance and activation. So our lymphocytes and our antigen-presenting cells are the white blood cells. And it kind of makes sense. So this is going to be lining our portals of entry. So our respiratory tract, digestive tract, genitourinary tract. And it kind of makes sense. If you're protecting a house, so think of the human body like a house, you put locks and bars on your doors and your windows, right? So that's the part of the house that needs to be protected. So mucosa-associated lymphoid tissue is basically that. So it's going to protect against entry. In the small intestine, particularly in the ileum, so the most distal part, there are these little white aggregates, so just deep to the mucosa, and they're called Peyer's patches. And here it's showing you lymphoid follicles in those Peyer's patches. And so what's happening here is there's a special kind of cell called an M cell that's just always going to be kind of sampling all of our food, because the food we eat is not sterile. Just sort of sampling that food, and the idea that any pathogen we've ingested, that we're going to mount our immune response right there before it gets any deeper. So immune response to ingested substances. So the small intestine are right here in the middle, and then we go small intestine, large intestine. So right here at the junction between the small intestine, the large intestine, you got this first part of the large intestine called the cecum. That's where you'd find the appendix. And the appendix is a blind-ended sac, and it is lymphoid tissue. The thing is that since it is narrow and it's long, when this lymphoid tissue gets activated by a pathogen and it responds through lymphocyte proliferation, so think like your lymph nodes swell, the appendix really can't swell. So it doesn't swell well. Oh, yeah, I didn't mean to say it like that, but yes, it doesn't swell well. So if the appendix is inflamed, it's called an appendicitis. And the risk is if this ruptures all of the bacteria in the colon. The colon is where our feces form. So yep, poop bacteria, a whole lot of it. So if this ruptures, all those bacteria get out in the peritoneum, and then you get peritonitis, sepsis, and you can get death. So it's really, you can live without an appendix, so it's just easier to remove the appendix than risk peritonitis. tonsils are going to protect against nose and mouth entry. So there is a pharyngeal tonsil. So this is the nasal cavity. This is the upper part of the throat, if you will. And then we also have, these are the palatine tonsils. Oh, look at those. Those are horribly angry and inflamed. And then at the base of the tongue are the lingual tonsils. So what tonsils are, they're just folds of lymphatic tissue, and they make what are called tonsillar crypts. So you see this big, deep invagination is called a tonsillar crypt. How fun, that makes them different. Deep to those crypts, these are all lymphoid follicles. So once again, it's sort of like inviting things in order to activate the immune system before it goes any deeper into the body. And so it's sort of, yeah, it's kind of playing a little dangerous game there, but just protecting against nose and mouth entry for both air and then also food. So things get stuck in those crypts, and then our immune fighters are just waiting right there. Okay, so this was the first episode. I'm going to send out a Kahoot challenge for you to help you make sure that you got this information. As always, I'm here for you if you have any questions. You have an awesome